Hi, everyone. I am Lacey Corset, and welcome to our first ever Gold Talk. If you're wondering what is Gold Talks, well, it's just like what it sounds. It is a virtual discussion where we dive into the research, the findings, understanding what is COL4A1, A2, and answering questions and educating the community to better understand this mutation. And we have an amazing discussion for you today. We are talking all about the introduction to Gold syndrome. And in this discussion, we will touch base on what is Gold syndrome. We will answer the top 5 most commonly asked questions. We'll get into diagnoses and resources and so much more. But first, let me introduce the man of the hour, the leading expert in Gold syndrome, as well as the founder of it, Dr. Doug Gould. Thank you so much for being here today.
Thanks for having me.
Yay. We are so honored that you took some time from all your research and your lab to come be a part of this discussion with us. Right off the bat, I wanted to ask, you are a doctor of science, not a doctor of medicine, correct?
That's correct. Yeah, I have a PhD in medical genetics.
Yes. So there will be, you are not able to do any medical diagnosis or treatments or treatment plans or individual treatment plans. This is all just talking about the science and the research that we found so far in this mutation.
Yeah, that's right. I have a basic science lab, and we do basic research, and we've been working on this for a long time.
So our audience would love to know if you could give us a little information about when and how you found this mutation.
Yeah, so a little bit about my background. I'm from Alberta, Canada originally, and during graduate school, I was studying eye development in the human genetics lab, and then moved to Bar Harbor, Maine, to the Jackson Laboratory to continue studying eye development. And while I was there, sort of a back burner project was to find new mouse models of defects of eye development, so ocular dysgenesis, and how they might cause glaucoma. And so we found this new strain of mice that had a mutation. We didn't know what the gene was, but we knew that these mice had ocular dysgenesis. And so using sort of old traditional gene mapping approaches, identified that the mutation was in collagen 4 alpha 1. And mutations hadn't been identified before in mice or humans for collagen 4 alpha 1. And so in the course of those studies, we recognized that these mutations didn't only affect the eye, that they really can affect any organ, and principally, in fact, the blood vessels in the brain, the cerebrovasculature. And so I got a fellowship from the Canadian Stroke Network to understand the role of collagen 4 alpha 1 in cerebrovasculature.
Wow. And this mutation is so new, isn't it, in terms of just the research and in the medical field as well. And you do have your own lab with the University of California, is that correct?
Yeah, that's right. So my former postdoc mentor, Simon John, is very interested in glaucoma. And so as this was sort of taking a different path, he was very generous and allowed me to take this project. And in 2006, I came to UCSF to start my own lab. And that's really, the entire lab is dedicated to just studying collagen 4 alpha 1 and collagen 4 alpha 2.
How amazing. And how grateful we are that you have a whole lab devoted just to this mutation. If you take a look down at the bottom right of your screen, you will see a QR code. And if you would like to help further the research that Dr. Gould and his team is doing, please feel free to scan this QR code and it will go straight to his lab website where you can donate and further the research. And we'll also bring it up later in this discussion. Now, with this being so new, and our audience and the majority of them, I feel like have just discovered this mutation within the last few years. I want to take a step back for a minute and have this discussion be as if I'm one of those maybe new adults, or I'm one of those new parents where my child just got diagnosed. And oddly enough, I was a couple of years ago with my child. And with all of the diagnosis and trying to figure this out and the questions after receiving and those genetic results, what is Gold syndrome?
Yeah, so, and I'll give you a little bit of background there too. So, it was in 2005 we found the first individuals with mutations in collagen 4 alpha 1. And at the time, it was this very rare, a rare genetic cause of a rare disease called porencephaly, which is these large cerebral cavities in the brain that are secondary to intracerebral hemorrhages in utero. And so those were the first individuals identified, and people. It wasn't very frequent that these mutations were found, because in those days, to identify which gene might underlie a condition that you thought might be genetic, you had to have a very good idea of what genes might be involved. You could only really sequence one or two genes at a time. And so in the early days of this, and 2005 to 2010, let's say, there weren't very many people identified. But more recently, with the widespread adoption of exome sequencing or genome sequencing, where you don't need to guess what gene might underlie a potential genetic disorder, you can just sequence all of them. Now new patients are being identified all the time. So, I get contacted by one or two new people every month. There are, over 500 or so now at last count. So, but what is it? So it's really a multi-system disorder. It can affect any organ. So I mentioned the cerebral vasculature, and I mentioned ocular development. Or ocular dysgenesis, but it really can affect any organ. Most commonly, the cerebral vasculature, and this is also probably the most consequential part of the syndrome. But about a third of the individuals have defects of the front of the eye, developmental defects of the eye, which can also predispose to glaucoma, which is a blinding condition. About 20% of the individuals have renal involvement. About 15 or 20% of people have skeletal muscle involvement. And so those are sort of the main organs that, are typically involved. But again, it really can affect any organ in the body. And so, in terms of, that's sort of the phenotype side of it. So the clinical picture, but what is it in terms of the molecular biology? Mutations in collagen 4 alpha 1 or collagen 4 alpha 2 are what causes Gould syndrome. And so to take a little step back, collagens are a major family of extracellular matrix proteins, and collagens, that family of collagens, make up about 30% of the proteins in your entire body. Now, there's 28 different kinds of collagens, and these are the type 4 collagens, okay? And of the type 4 collagens, they're encoded by 6 different genes. So collagen 4 alpha 1, collagen 4 alpha 2, and then collagen 4 alpha 3, 4, 5, and 6. Collagen 4 alpha 3, 4, 5, and 6 are much more spatially restricted, and it's been known for, say, 40 years that mutations in some of those genes cause something called Alport syndrome, which mainly affects the kidneys. But collagen 4 alpha 1 and collagen 4 alpha 2 are present in every organ. And so that's why, mutations in these two genes can cause this multi-system disorder. Now, the genes are turned into proteins. They encode a protein. And so the collagen 4 alpha 1 gene makes the collagen 4 alpha 1 protein, and the collagen 4 alpha 2 gene makes the collagen 4 alpha 2 protein. And these two proteins find each other inside the cell, inside a special compartment of the cell. And you have 2 collagen 4 alpha 1s and 1 collagen 4 alpha 2. And they find each other, and then they braid together like a little piece of rope, right? So it's 3 strands braided together, and then that little piece gets secreted from the cell, and once it's in the extracellular space, they assemble into something like a fishnet, and then that creates these flexible sheets. And these flexible sheets can sort of partition different cells in a tissue, but they can also have really important biological functions. So they can transmit information to cells. They can tell them where they are. They can tell them to divide. They can tell them to do all sorts of things. So they're kind of like a scaffold, but they're like a signaling scaffold that can instruct cells and really organize tissues.
So what diagnoses do you typically or commonly associate with Gould syndrome that you've seen in your research?
Yeah, so I mean, on the slide right now is from a survey from a global registry that the Gould Syndrome Foundation had hosted. And so this is a family-based or individual-based or patient-based self-reporting. And so, the major things that came out of this are, certainly the cerebral vasculature is one of the major complications and consequences, and also seizures and infantile seizures were really a major concern of the field. But again, I think that this underscores that that the mutations again can kind of affect multiple organs.
Yes, and you can see, you were talking about how this mutation primarily affects, bleeding of the brain. You have cerebral palsy up there at the front. You also, of the eyes, you see the vision impairment as well. You talked a little bit about the kidneys, which you can see down farther below. And no, I do understand that these findings were just for pediatric patients, but could this data also in these diagnoses be seen in adults?
Yeah, and so one of the real calling cards of this syndrome is also that it's highly variable, which again was one of the reasons why it took so long to be identified as a syndrome. That no two patients look alike is maybe a little bit of an overstatement, but it captures the essence of it. That individuals can look wildly different. Even siblings in the same family can look very different from each other. So already, there's a wide variability. Now, it's common in genetic diseases that the first individuals identified are going to be the most severely affected, or the early onset severe cases. And then you might identify the gene. And then as time marches on, you start to identify individuals who have milder or atypical cases, for example. And so, following that same mold, the first individuals that we found with mutations were, like I mentioned, had poor encephaly. There are very few familial cases where it's inherited through sort of 3 generations. So you have adults who carry the mutation and are relatively healthy. So I think almost by definition, the adult cases tend to be on the milder side, but there's also an ascertainment bias there. And so I think, again, the take-home message is that if you are identified with the mutation, even if you don't manifest symptoms in other organs, you should really have testing to get baselines for these things.
Yeah, and that, which is so important, and we'll bring that up in a couple of slides. Now, with the diagnoses, we also have, commonly associated specialists, correct? This here that you're looking at is a poll, and I want to give a special thanks to our COL4A1, A2 support group page. I went on there, we created a poll, and we asked you, our audience, what specialists that you had or that you were currently being treated from. And thanks to y'all, we were able to collect this data, and this data was collected on October of 2022. We had probably about 80 to 90 participants and a total of 707 votes. So here is your top 10, which is both pediatrics and adult specialists that are commonly associated with Gould syndrome. Now, with such an influx of tons of newly diagnosed adults and newly diagnosed parents with children, we get some very commonly asked questions. And thank you so much for addressing these. We have presented with our audience the top 5 most commonly asked questions. So let's hit it with the first one. What is COL4A1 versus COL4A2 versus Gould syndrome? Because we get this one so much.
Yeah, no, thanks. And so, like I mentioned, I think, every couple of weeks or at least once a month, I have phone calls or Zoom meetings with families or parents of people who've just been newly identified, and many of them, of course, have the same questions. And so this is clearly one. So what's the difference? Especially, and I should stress this, especially people with COL4A2 mutations, because these are less common. For reasons that we don't quite understand, collagen 4 alpha 1 mutations are about 10 times more common than collagen 4 alpha 2 mutations. And so to answer this, I mean, the quick answer to this is that, the syndrome is really what, the clinical manifestations and how the patient presents, and collagen for alpha 1 and collagen for alpha 2 are the genes that might have a mutation that lead to that syndrome. But I'll just back up a little bit and sort of nerd out, tell you a little bit about genes and proteins. So as I mentioned, get my pointer. Collagen four is encoded by 6 different genes. So this is collagen for alpha 3 and 4 over here, and 1 and 2 in the middle, and 5 and 6. And so these genes sit on different chromosomes, but in pairs, right? So collagen for alpha 1 and collagen for alpha 2 in humans sit on chromosome 13, and they sit head to head, where they're transcribed or they're read in opposite directions. And then you get the collagen for alpha 1 protein and the collagen for alpha 2 protein. And that's what's shown on the bottom here. In red is collagen for alpha 1, and in blue is collagen for alpha 2. And the protein has 3 major domains. On the right side here, this NC1 domain is sort of a globular domain. So it forms this ball with a bunch of different features on it. But most of the protein is this long triple helical domain. And this domain is what makes a collagen a collagen. Okay, there's 28 different kinds of collagens. And the defining feature is a repeat of glycine XY. So glycine is an amino acid, right? Proteins are just chains of amino acids. And if you have glycine, something, glycine XY, gly-XY, 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 over and over and over again, you're a collagen. And you need glycine at every 3rd position, because glycine's the smallest of all the amino acids. And so it can fit into the core of that braided rope. So it's tiny and fits into the core. And so if glycine gets changed to anything else, it now has a little bit of a side chain, it's called, and it can mess up how well that rope braids. And the cell can recognize that it's not formed quite properly. And the cell has a quality control mechanism, so it sees that as misfolded, and it doesn't want to secrete it. And so this is just a little bit of a different view. This is showing, a cell on the left-hand side, and this cell is making collagen, okay? So again, in red is collagen for alpha 1, and in blue is collagen for alpha 2, and then they braid into these little ropes, and then they get packaged and then secreted from the cell. And so again, the major consequence with these mutations, and the most common type of mutation, is any one of these 300 or so glycines, any one of them gets changed to something else. This braided rope doesn't fold properly, and so it doesn't get secreted. Well, what does that mean? I told you earlier that once it gets secreted from the cell, it forms like a fishnet kind of flexible sheet. Yeah. And that those flexible sheets have functions. And this is just showing, hypothetical examples of how a different cell is responding to that collagen, okay? And so, this is sort of getting to what the basic mechanisms are. If the protein doesn't fold properly, you might have too little collagen in the basement membrane, or you might also, some of the mutant collagen can get out, and when it gets out there, it can cause problems. And so that's sort of the simple way to think about the disease mechanisms at the cell and tissue level.
Now, I do want to touch real fast on the COL4A1, A2 versus Gould syndrome. Is it correct that in terms of most of the research out there, and even in the medical field, you will still see it as COL4A1 or A2, and Gould syndrome is still kind of a new name that, if I'm correct, is just recently, within the last 2 years, we started using?
Yeah, so it really came from when the foundation started, and they were trying to put a name on it that wasn't a long alphabet soup, which is what we'll get to in a minute. So it gives it something that people can identify with, and it also sort of simplified things in that there have been other names that are sort of clinically based. One of them, for example, that you might see is Hannak syndrome, and some, and others. These were all based on different clinical descriptions, which can be limiting because not all individuals have the same clinical description. So, do I have this disease if I don't have renal involvement, or do I have this same syndrome if I don't have muscle involvement? And so it really kind of gave it an umbrella term that people could rally around, and it was sort of driven by the foundation trying to come up with a good handle.
Right. I think of it kind of like, instead of saying, oh, well, I have an extra chromosome on chromosome 21, people just say, well, I have Down syndrome. So instead of saying, oh, I have a mutation on COL4A1 or A2, I have Gold syndrome. And I will put a little plug in there for our audience who is watching. Don't be surprised or worried too if you see, like, in your child's medical records, or if you mention this to a doctor and they still refer it to COL4A1 or A2. I think Gold syndrome is slowly getting out there, but it's just taking a little more time for people to catch on with it. Now, our second most commonly asked question: how do I interpret the letters and numbers in the mutation? I remember when I got my child's genetic paperwork from the genetic lab, and they said, mutation COL4A1, and then it was like this alphabet soup with a bunch of numbers mixed in. Like, what in the world does that mean?
Yeah, no, yeah. And this question comes up all the time. And so again, I'll just step back a little bit and give a short biology lesson. But genes sit on chromosomes, and chromosomes are just a string of a G, A, T, C, T, C, G, A. You have these 4 nucleotides over and over and over again. But the genes are sort of interspersed, where parts of the gene, which is called an exon, my cursor's not working great. They have parts of the gene that are going to be turned into a protein interspersed with introns, parts of the gene that won't be turned into proteins. And so all of the exons have to get spliced together. And once all of these exons get spliced together, now you've got the continuous string of nucleotides, or AGs, Ts, and Cs, that are actually going to get turned into a protein. And once you have that, that's the coding part of the DNA. Coding meaning it's going to be translated into a protein. And so that's numbered from nucleotide number 1 up to whatever it goes to. And so when you get this alphabet soup, I chose one example. This is actually one of the most commonly occurring mutations, so I chose it for that reason. So, of course, first it says COL4A1. That tells you which gene it is. This is COL4A1, not COL4A2, but in others it would be COL4A2. Then C dot, that tells you that this is just the coding region now. And then this is the number of the nucleotide that's changed. So in this more common variant, nucleotide number 2263 should have been a G, and it is instead an A. So this tells you the nucleotide change and where it is in COL4A1. And then the next part of it is P dot. And now that tells you, we're talking about the protein. Okay, so that coding DNA gets translated into a protein where every 3 nucleotides are a code for 1 amino acid. Okay, and so the protein is just a chain of amino acids. And so this tells you that in the protein, this is glycine at position 755 got turned into an arginine. Okay, so it's very similar. And every amino acid has a single letter code, but they also have 3-letter codes. So you might also see this written as gly 755 arg. But again, it just means in the protein, glycine at position 755 got changed into an arginine. So this whole string tells you the gene, the nucleotide position in the coding DNA, and the amino acid of the protein, its position and what it was changed to. And as I mentioned, almost all mutations that we know to date are mutations in these glycine residues that have to be present at every 3rd amino acid.
Now, say for example, what you have here is my child mutation or variant. Would you say that it is, if he was the only one, is that pretty common for something that is newly diagnosed?
It is. I mean, I would argue that we're still very early in the game, and that eventually, almost, my bet is that almost all of these glycine residues, we will find somebody who has a mutation in one of them. That's probably the way that it will play out. But these are very early days. And so most people, when they find a mutation, it's never been found before. As I mentioned, I think it's around 330 different glycine residues. And so, eventually we'll get to saturation where each of those there will be somebody who has a mutation in it. But at this stage, most people are identified. It's never been found before. Now, this particular mutation that I chose for this example, even when we published a review article about 4 years ago, this mutation had been independently identified in, I think, 7 different people so far. So there is some recurrence, but at this stage, that's still less common than it being basically brand new.
So if you or your child is the only one who's had this gene variant, don't panic, basically.
Yeah, exactly. It just means it hasn't been found before. This is the first time.
Great. Thank you so much for clearing up that question. I know that's one that's commonly asked to the follow-up of the alphabet soup that you just explained. Most commonly asked question number 3, what is the difference between inherited and de novo?
Yeah, so inherited means that the mutation was passed down from one of the parents, and de novo means that it didn't. Neither parent had the variant and that it was brand new. It occurred for the first time in that individual. And so, for most cases, about 2/3 of the time, the mutations are de novo, meaning neither parent carries the mutation. About 1/3 of the time, it is passed down from one of the parents. And so, a lot of people also ask, well, how is it that you have a seemingly healthy parent that passed on this variant to potentially a severely affected child? There are a number of explanations, but probably the simplest, but there's not a lot of data on this, so this is some speculation, is that there's something called genetic mosaicism, meaning that parent probably or may not carry the mutation in every one of their cells. So they're mosaic, they're genetic mosaic. Some cells have the mutation, some cells don't. And this is, common with some genetic diseases, but especially with diseases of extracellular matrix proteins, it's much more possible for the cells that don't have the mutation to secrete normal protein and compensate for those cells that do have the mutation. And so that's one way where you can have an individual who's mosaic that seemingly doesn't have any phenotype or is seemingly healthy because some of their cells are able to compensate for the others. There's really only a very small number of families, maybe 5 or so in the literature, that have 3-generation inheritance, meaning that the person in the middle generation definitely carried the mutation in all of their cells. But again, most of the time, 2/3 or so, these are de novo mutations, so neither parent carries it.
Now, why, I know we talked about it, kind of like the umbrella of it, why does every person seem to be affected differently? I know on the social media pages, we've had some that just have a few kidney issues, all the way to some having severe developmental delays and cerebral palsy and kidney issues. And so why is there this huge umbrella when it comes to this mutation?
Yeah, it really is, again, like I said earlier, that's really one of the calling cards of the syndrome, is that it's highly, highly clinically variable. You might not guess that 2 individuals with these mutations have the same disease. And this is even true with people who have the same mutation. I mentioned earlier that this glycine 755 arginine has been recurrent, but the people with that same mutation don't necessarily have the same phenotypes. But there's a lot of different things, a lot of different factors that can contribute to why a mutation might present differently in different people. For one, so there's collagen for alpha-1 and there's collagen for alpha-2. So far, it seems like they're pretty equivalent, but there might be some differences there that we just don't understand yet. From work in mice, we know that different mutations behave differently. So a mutation in one part of the protein is more likely to cause more severe disease than a mutation in another part of the protein. But so that probably plays a role. but I don't think it has, this is sort of jumping ahead to the next question. It doesn't necessarily have prognostic value. You can't necessarily say, oh, my mutation's over here, so I'm going to be milder or more severe. It doesn't quite play out so cleanly. Again, from work in mice, we know that the genetic background can have an effect. So, for example, two mice that have the exact same mutation but have different genetic backgrounds, so they're on different strains, can have very different phenotypes. So one is very, very mild, another one's very, very severe. So we know that how the mutation interacts with other parts of your genome or other genes in the cell can influence the way that it plays out. And then, of course, there's environmental factors. So things like high blood pressure or head trauma or birth trauma. And then some of it just comes down to sort of stochastic factors. So you might be at an elevated risk for stroke, but when that stroke happens could play a large role in the outcome, or where in the brain the stroke happens can affect people differently. So there's a lot of different things that come into play. Part of them genetic, part of them environmental, and that's sort of the answer.
Now, I'm sure you get this question all the time, and I know this was one when I first got my child's diagnosis. But what's the prognosis? Do we know the outcome of this?
Yeah, we don't. And part of that is also related to the last question, where in any one individual, we certainly can't say what the prognosis might be. And this is actually a problem for us. This still is sort of early stage, and we need a lot more research in this. We need to know what's the natural history of the disease. What's the, what are the likely outcomes? And this is where we do need people involved, and we need to get people involved in the research and get as much clinical history and clinical background as we can. Imagine that one day we do come up with a drug or some intervention that we think might work. Well, in order to show that it works, we have to have solid endpoints, and those endpoints need us to know, what the likelihood of any particular event might be over, say, a 5-year time period. So the prognosis right now is really it's up in the air, in part because it's so variable, and in part because we just don't have the data yet.
Yeah. I'm newly diagnosed. My child's newly diagnosed. I figured out what it is. Thank you. I have my top 5 questions answered. Now, where do I go from here? And are there labs? Are there scans? Are there annual monitoring that's recommended with it that you've seen in your research?
Yeah, so I mean, again, I think it comes down to sort of having a full body workup, making sure that talking with your doctors. Again, the cerebral vasculature is sort of the major concern for most people. So talking to your specialist and doctors about stroke prevention, having MRIs and angiograms, just kind of looking at the slides here. Again, this sort of second most common feature are ocular manifestations. So monitoring intraocular pressure, which if the pressure gets elevated can lead to glaucoma. Looking at the retinal blood vessels, because the retina is just kind of an out pocket of the brain, and so looking at retinal vessels can help give an idea of what might be going on in the cerebral vessels. The heart, so having a cardiac echogram. Kidneys, as I mentioned, are involved in about 20% of patients. So having kidney and liver ultrasounds or CT, looking for hematuria. I mentioned that there's skeletal muscle involvement, and so looking for creatine kinase, which is sort of a marker for muscle damage. And then establishing care with the geneticist, and then finally sharing the test results with close family members, so parents or siblings or children, certainly, so that they can speak to their providers about the benefits or risks or pros and cons of having genetic testing done. And then, in terms of management, so individuals that are at a high risk for hemorrhagic strokes or cerebral bleeds, and so it's recommended that they stay away from antiplatelet or anticoagulant treatments and intravenous thrombolysis. So these are things that can exacerbate a risk of bleeding. Anything, you want to avoid head trauma. So sports that put you at high risk for head trauma, or prolonged excessive exercise should also be avoided. And then in pregnancies where it's known that the fetus carries a mutation, then cesarean section should certainly be considered to hopefully also alleviate any sort of trauma to the head during vaginal birth.
Okay. And when it says antiplatelets and anticoagulants, we're talking about medications like NSAIDs, or non-steroid anti-inflammatory drugs, like your Motrin or your ibuprofen, your Advil, Aleve, and then also like your anticoagulants, like your warfarin, that would thin out your blood. Is that right?
Yeah. So any of those things that can sort of lead to increased risk of bleeding.
For those of our audience who are still watching and want to receive more resources, there are more resources out there. Take a look. If you haven't had a chance, go to the Gould Syndrome Foundation website, and under there, there's a tab that is for resources. And it is a wealth. We're not going to get into it, but there's a lot of different resources in there that you can take a look and utilize. So please do so. And not only do we have the foundation website, but here I listed it, the Gould Syndrome Foundation website, but for our audience in Italy or in Europe, they also have their own foundation, which I listed. And not only for the resources do we have websites, but we have support pages on social media. We have two. The first two are support pages. The third is for the Gould syndrome. That's a support page on Facebook. And then, of course, we haven't forgotten our friends in Italy and Europe. They have their own one as well. Just a quick reminder for the social media pages, for the support pages, they are not used to treat. They're not used to diagnose. So if you do receive feedback from that, please understand that that is support. It's not medical advice, and to always consult your personal physician. But to help you utilize these resources a little bit better, don't forget, you can go to the Files tab under More, and there are some resources and documents there that you can take a look at. Especially, I'd recommend that for our newly diagnosed individuals or family members. And what is so cool about this is the search bar as well. Say, for example, all of a sudden your child has cataracts. You can go up to that search bar, you can type in cataracts, and it will pull up all the previous posts and discussions associated with cataracts. So it will just, and as you put it in there, it'll give you a wealth of different information, and you can see what other parents or what other adults have said in terms of having this mutation in cataracts. So this is a great resource and tool to use for support and to see what other people in the community have experienced as well. Now, this is something I'm really excited to talk about. You have your lab, which is associated with the University of California in San Francisco, and I really don't know much about it. Can you tell me a little bit about, like, how big is your lab, what you guys are focusing on?
Yeah, so the lab sort of fluctuates between maybe 8 and 10, 12 people. And there's a combination of senior researchers and postdocs and junior technicians. And, like I said, our focus is really, we have multiple projects, but the common thread running through them is trying to understand biological functions of collagen for alpha 1 and collagen for alpha 2. I showed you the slide about sort of the tissue level view of it. So we're trying to understand things from very, very basic science perspectives. For example, how is collagen made? The biosynthesis, how is it packaged and secreted? How does the cell recognize when collagen's not folded properly? Because we know that if we can promote secretion of the mutant proteins, that often that seems to be beneficial. So that looks like a pathway that might have some potential therapeutic value. But also, once collagen gets into the basement membrane, what are its biological functions? We just don't know these things. It's a fundamental and primordial protein. So the type 4 collagens are over 750 million years old. They're throughout the entire animal kingdom, but we really don't know, what does it do? What are all of its functions once it's in the basement membrane? And of course, once we know what those are, those things might also present themselves as therapeutic targets. So it's really sort of discovery-driven science with an eye towards how might we be able to translate these findings therapeutically.
I do want to let everyone know there are research papers available, research papers that are free. You can go to Dr. Gould's lab. The link is here, and you can look at the published research papers that he has put together thus far. And you can go to the Gould Syndrome Foundation website, and it will be under research. One of the things I did want to mention is when I first started looking into the research papers when my child was diagnosed, and I had a chance to ask you, I asked you which one of the research papers would be the best one to hand to a doctor or a specialist who perhaps hasn't heard about this, or maybe has heard about it but doesn't know much about it. Because right now, with it being so new, that's pretty typical. I've found that sometimes we'll see a new specialist or we'll see a new doctor and they've heard about it, but they may not know a lot about it. So you have recommended this research paper entitled Genie. Can you tell me a little bit about why you recommended that one?
Yeah, I mean, at the time, I think that one sort of gives a nice summary of sort of the genetic landscape. It lists all of the mutations that were known in the literature at the time and their positions, and broke down some of the statistics of the types of amino acids that are changed and what they're changed to and where they are. So a lot of this sort of molecular biology, but also a summary of the phenotypic outcomes. Yeah, and it's a few years old. I think probably we would do well to get another review out there. But I think it's a nice sort of high level overview for somebody just starting to that's interested in getting an understanding.
Right. I know I've taken it and I've printed it a couple of times, and I keep a little stash. So every time we get a new specialist, and I kind of get a feel for, have you heard this before? Do you understand it? And even if they have, I still hand them the paper so they can read what's been published out there and get a better understanding of it. And it's been such a great help.
Okay.
Now, I know for our community, for our parents with kids with diagnosed, even for me as a mom with a child who has Gould syndrome, we're all trying to figure out how can we further the research? How can we help? How can we move things along so we can get more education, more understanding, more answers to the question? And it really all comes down to donating and fundraising. Is that right?
It certainly helps. I mean, having families and patients and parents involved is enormously valuable. I mean, like, as we said, my lab's a basic science lab, and my interactions with people has helped guide some of our research. So it helps us understand the challenges of the families better and understand the things that are important to the patients. And also getting families involved in research is important. As I mentioned, we need to know the natural histories. We need to understand the clinical aspect of the disease better. But yeah, I mean, so raising funds is enormously helpful. It allows us to hire more people or keep people longer, or seed new projects that we think are interesting but don't just yet have federal funding yet, or it helps us continue projects while there's gaps in grant funding. And so research funding is the lifeblood of our work and of the lab, and it's enormously helpful. So we appreciate all this.
I was going to add, the research is where everything starts. You have the funding to help with the research. The research helps with publishing research articles, which then go to the doctors and the specialists, and they are able to interpret to thus figure out how to help each individual with this mutation. So it's really a donation or a fundraising process that helps everyone down the line. We're talking specialists, doctors, scientists, and especially our community, our Gould syndrome community. So if you get a chance, here's our QR code. You can scan it. It will take you straight to Dr. Gould's lab. It is not a GoFundMe page. It is directly associated with the university that he works with, and it is a great opportunity to help further the research and help our community. If you need a reason to start donating or doing fundraising, our Gold Syndrome Awareness Day is April 12th, and we wear yellow. So it's an awesome opportunity to start thinking with the new year coming, hey, maybe I want to put together some kind of fundraising. And can you talk a little bit about how easy this can be?
Yeah, so just to highlight one recent example, it's just beautiful. So this is the Woods family in Missouri, and for their son's first birthday, they did Landon's first lap, and they got friends and family together and did a walk, and they raised, I think it was over $12,000. So it's heartwarming, and you know, we really appreciate it, and it's outstanding. And last year around Gould Syndrome Awareness Day, we also had a couple of large donors. An individual gave over $100,000 and matching funds towards other people who were donating, and I can't tell you how much you know, help that gives the group. So we appreciate it all. Thank you.
Yes, thank you guys to all who have donated, who've already been out there and actively helping fund this cause. It's such a wonderful cause. Now, don't worry, we're not going to wrap it up just yet. I do want to thank you, Dr. Gould, for being a part of this and taking time away from your lab to really help, especially our newly diagnosed community in this discussion. But we're not over, and Gold Talks is not over yet either. We want our audience to help us with our next Gold Talk topic. So if you have follow-up questions to this discussion or general science questions about Gold syndrome, we would love to hear from you. Please submit your questions to goldtalks@gmail.com with the subject line Gold Discussion by May 5th, 2023, and we'll see you guys soon. Again, thank you so much for being a part of this discussion, and we look forward to seeing you guys soon.